means we're recording. Okay, here we go. We covered chapter 1 the other day. We do not cover chapter 2 and 3 in class, but you should have covered them. They are recorded. They are up on Rock Creek Bio Group, and they're on D2L as well. So you can watch them at home on your own in the comfort of your own house with your pajamas on, if you like. What's this a picture of? Anybody know? It's one of the cooler pictures I ever took, I have to say. Yes? Is it a plane taking off? Yes, it is. It's a helicopter taking off. This is a United States aircraft carrier. This is the USS America that I was stationed on for a few months in the Mediterranean. And we did most of our flight ops at night because the pilots were so good that they didn't really consider it a challenge to take off and land during the day. They really wanted to practice taking off and landing when there was about 40-mile-an-hour winds and the ship was going up and down and up and down, and we were in 30-foot seas, because that's when they could really prove they had the right stuff. You guys ever read the book, The Right Stuff? It's about the original astronauts. It's a wonderful story. They were all Navy pilots, just about. Navy and Marine pilots became the original astronauts. So this is the front of the aircraft carrier. I'm actually sitting in the captain's seat, taking that picture with a long delay. And this is there's two lights on the helicopter, one underneath it and one on the top of it. And they're those old rotating kind of police car lights. And every time they would rotate, you could see the light bright. It would make another. Anyway, that's all. How do we learn? Well, if you hear it in lecture, I think I've gone over something like this that Confucius said. This is from a study on that. If you read it, 10%. If you see it in an audiovisual format, you might remember 20% of it. If you see a demonstration of it, you might learn 40% of it. If you have a discussion about it, you might learn 50% of it. If you do a hands-on project, you might learn 75% of it. But if you teach it, and if you teach it to your lab partner, or you teach it to those 3 other people in the lab, or you teach it to your friends at home or your husband at home, you will know 90% of it. Because you don't tend to teach things, and I don't either, unless I really know what I'm talking about. I really don't stand up here and BS you. I really do know what I'm talking about here. And that's because I've taught it a few times, and so I've probably got close to probably 95% there instead of 90. But I think it's the point to be learned there is the best way for you to learn it is for you to teach it. So keep that in mind and use all those opportunities that you have in lab and in open lab to teach others, and then you will really learn it yourself. Okay, so we're going to talk about tissues. Now, what's a tissue? We talked about that the other day. We said there was cytology and there was histology. And cytology was the study of individual cells, and histology is the study of tissues. So like when we would do a pap smear, you're just scraping off a few loose cells and you're looking at them under the microscope. If you do a biopsy, whether that's a needle biopsy where you fill up the needle itself with tissue, or whether you actually take a chunk of material out of somebody, like on a breast biopsy, that's tissue. Okay, it's a chunk of tissue, and that's what we will be studying in A&P mostly. In fact, we don't really do any cytology except for the blood, where we do individual cells in the blood. So there's 4 major types, and you can learn that right up front here. Number 1 is epithelium, and what epithelium means is it's the outermost layer or the innermost layers of anything. So your skin is an epithelium, your inside lining of your mouth is an epithelium, the inner lining of all your blood vessels, the inner lining of all of your intestines, the inner lining of everything is an epithelium, whether it's the outside lining or the inside lining, okay? And we're going to spend most of our time talking about the different epitheliums, because that's the best way to identify stuff, really. The second one is connective tissue. Connective tissues are just what you think. They're the stuff that connects everything together. They fill in the spaces between the other type of tissues. So those are things like fat and what we call connective tissue. Blood is a connective tissue. Lymph is a connective tissue. Those are fluid types of connective tissues. Third one is muscle. And you're going to learn that we have 3 different kinds of muscle, and you need to be able to tell the difference between the 3 and know the difference in how they work. And the fourth one is nervous tissue. The different cells that make up our brain and our nerves, they're quite different. They have only one job, and that's to carry electric sparks around, electric shocks. And so we'll learn about them as well. Okay? Well, there they are. Connective tissue stores energy. Fat stores a lot of energy. Transports materials. That's like blood. Bone, by the way, bone and cartilage are connective tissues. They support things and they fill in all the spaces in between. Muscle tissue is 3 types. Skeletal muscle, which is all the muscles that we have that we can control, that move our bones and all that jazz. One special kind of muscle we have is heart muscle, which is designed for the long run, so to speak. It never stops. It has its own system of pacemakers and so forth. We'll learn a lot about heart muscle. And then in all the walls of all the hollow organs, the arteries and the veins, in the walls of the bronchial tubes in your lung, they have a lot of muscle that can either open them up or close them down. We can open up blood vessels in order to allow more blood to flow to one area. We've got muscles that will close it down to send less blood to certain areas. So we'll talk about that. That's called smooth muscle. And then last one is neural tissue, which honestly is really simple. We won't talk about it for about 10 minutes. Okay, so let's talk about epithelial tissue, and that's the first one up here. And before I even get over defining the word, which I've already done really, it's layers of cells that cover both the internal or the external surface. So even though it's called epithelium, which you might think is on the top or the outside, it also means the inner lining of things too. Epithelium is the type of tissue that formed all of our glands. And that's kind of an odd thing if you think about it. Why would our thyroid gland or our ovaries or any of those other things, why would they have been formed from epithelial tissue, from stuff that was on the surface? Well, let me draw you a little series of pictures here. Maybe I'll go over here. And let's just say that the way it works is like this. It starts off like this. So this is the epithelium layer, the surface of something. And then it starts kind of building up and getting thicker right there. And then it goes to this, and it gets really thicker like that. And you've got a bunch of cells. And then it goes to this, and it kind of pinches off. And this set of tissues now becomes a gland. So say it becomes a thyroid gland, and it's completely separate from the skin or from the inner lining of the respiratory system, which is actually where the thyroid came from. So that is the process by which all the glands are formed. And just so you know that, because glands are all caused by this epithelia, we would call this process here invagination. It grows in and then pinches off. Invagination. All right, I'm going to have to keep up with my clicker today. So what are the characteristics of epithelial tissue? Well, first of all, I told you it was either on the outside or it was on the inside, so therefore it's always got polarity. In other words, it's got an apical surface, which is the top surface, the surface that opens to space, either the outside here, the outside of a blood vessel, or the inside of a blood vessel, whatever. That's called the apical surface. And then the basal surface is the inner surface of those same cells, and they were always attached to something. The thing they're always attached to is called the basal lamina, basal layer, okay? So just below every epithelia, a row of epithelial cells, which may be one cell thick or several cells thick, there is a basal lamina. That's a characteristic of epithelial tissues. Cellularity means that epithelia are always made up of cells. They aren't made up of anything else. It's always a cellular material, lots of cells with nuclei and all that jazz. They are avascular. Epithelia does not have any blood vessels running into it. That's what avascular means. No vascular tubes. So it has no arteries and has no veins. And what that really means is the way that epithelia survives, the way those cells survive, is they simply absorb things from osmosis or from other mechanisms of getting different things into them and getting the things they need, like sugar, into the cells and oxygen that they need, and getting carbon dioxide and so forth out. But it just is, the only thing they communicate with is the fluid that's around the cells, which we call it interstitial fluid. Now you may say, well, gosh, I've cut my skin and it bleeds. It's vascular. So what's the deal? Well, the point to remember is that the epithelia, the epithelial tissue, is just that top layer or two. Now in the skin, it can actually be 20 layers of skin cells but they have no blood vessels going up through there. How many of you have ever had a paper cut on your hand and it didn't bleed? Okay, see, that is a cut that doesn't go all the way through the epithelium. It hurts because there's nerve fibers in it, but there are no blood vessels. That make sense? And then the last one down there, I think it's the last one, is that epithelial tissue can usually regenerate. So if you bite the tissue off the inside of your cheek, it's going to grow back. If you whack a piece of skin off, it's going to grow back, close in from the areas around it. It can regenerate. It can make more of it. This is a generalized picture of epithelial tissue. It's kind of a combination of a bunch of different kinds of epithelial tissue, but basically, here's the points. There's a basal layer. This is the basal lamina down here at the bottom. This is the apical surface. This is the surface of the tissues. A lot of epithelial tissues have cilia on it. Cilia are these little things that sort of sit there and move all the time in order to sweep things out of the way, like on the inside of your breathing tubes. When you breathe in dust, you want all of those things to be moved up and out, up to your mouth where you can swallow them down, right? They go down into your stomach and you kill all the bacteria, and hey, that's the end of them. But you got to get them out of your lungs. You got to get them out of your breathing tube. So we have these cilia that help with that. Now, because this is just a generalized picture, a lot of epithelia cells have microvilli. And microvilli are like cilia, except they don't move. They just sit there. And what they do is, instead of it being straight, which wouldn't have that much surface area, it has all of these ups and downs, and that can triple, quadruple, quintuple the amount of surface area there is. So, for example, on the inside of your intestine, the inside lining of your intestine is covered with lots of microvilli like that, so it can absorb all the sugars and the proteins and stuff that are floating by in your intestinal contents. Okay? And that's the only function of them, is just to make more surface area for things to get picked up out of the surface out here and absorbed into the cell. The thing your book talks a lot about is these cell junctions, which are illustrated here, here, and here, I believe. And that's basically, just remember that epithelia cells are pretty much tightly attached to each other. So that makes sense. You need them to be tightly attached. Remember that epithelial cells are going to control what comes into your body, what comes into the cells of your body, and what goes out into the surface. I had a guy pushing me the other day. He was trying to sell me Advocare. You all heard of that stuff? He wanted to sell me all this stuff to get the toxins out of my body. And I said, well, that's interesting. What do you mean? And he said, well, all the stuff that's inside your intestine that's toxic to you, that's bad. We want to get that out. So we want to sell you this stuff that'll give you poops and get you, clean you all out and get all those toxins out of your body. Well, my answer to him is, wait a minute. The things that are in your intestine are not in your body. Remember, we have a pipe that goes from our mouth to our anus. And unless something gets absorbed across the membrane, it's just sitting in there. And yeah, it seems like it's in us, but it's really not. It's really not in our body. And our body is made to have all that stuff inside of the intestines. So go rock the boat, Keep them in there. You don't need to go having diarrhea for a week to clean yourself out when it's sort of made to be there, and it's just going to reform once you... Anyway. So epithelia really controls what's inside of your body. And when I say inside your body, I mean inside your cells, inside the inside of you. And what's on the outside is obviously on the outside. This illustrates a lot of epithelia cells secrete a lot of fluid and mucus and stuff. And as a result, they have active nuclei, they have lots of mitochondria where a lot of proteins are made. Those proteins are then packaged up in the Golgi apparatus. Remember, the Golgi apparatus is kind of like the FedEx of the cell. It packages things and sends them on their way. And so a lot of these Golgi apparatus will make little balls of mucus, for example. They'll work their way up here to the surface and be excreted. And that's why we get mucousy stuff secreted by the inside of our breathing tubes. That's why we hawk up a bunch of mucus if we get sick. We need mucus on the inside of our lips and stuff to keep everything sliding through there and keep it nice and wet. So all those epithelial cells that line our mouth are making fluids, just like the cells that line the pericardium and the peritoneum, the membranes we'll talk about some more, and we'll see in lab today. So what are the functions of epithelial tissue? Well, to provide physical protection. Believe me, your skin is the greatest organ you have. It keeps all the bad stuff out. It keeps all the good stuff in. Everything. It keeps heat in you. It keeps bacteria out of you. It keeps the fluid that's inside your body in you instead of out. And if you want to see the worst ever injury that a human being can endure, in my mind, it's a burn. It's a big burn. When somebody loses 50%, they have a burn on 50% of their body, and they lose 50% of their skin, just think about that for a second. Because what that means is that half of their body is now just weeping fluid. Fluid is just coming out of them. Not only water, but also proteins and electrolytes and salt and sugar and everything is just weeping out through their open skin because they lost that protective mechanism. And that's why burns are such a tragic injury and such a tough problem. So epithelial tissue, including your skin, provides physical protection. Believe me, the inner lining of your mouth and the inner lining of your small intestine provide protection against that stuff that you ate that they want to wash out of you. It controls the permeability. In other words, it controls what gets in and it controls what gets out. That's all controlled by the epithelium. It provides sensation. You know, if, how do you feel? I feel with my fingers, right? We have sensation fibers, nerves that pick up sensation in our skin, in our epithelium, that sense touch, sensation. They also sense heat, they sense cold, they sense pain, all these other things which we'll learn about. And the epithelial tissues produce all these specialized secretions. Now, a lot of it is just cells that are right in the epithelium on the surface, and they make mucus, like in our lungs. A lot of it is the endocrine, the glands that I mentioned at the beginning, the glandular cells. They all started as epithelial cells. So, what are some of the specializations that these epithelial cells go through? Well, one of the things is they do move fluids across the epithelium in both directions. They move them out of the cells to give us mucus and to give us moisture to line our cavities. But we also use it to absorb things, to get the food and get the sodium and get the potassium and get the sugar and proteins absorbed into our bodies as well. It has to come in through the epithelium. And producing secretions, which I've mentioned several times. And also these messengers. What's a messenger? What's another name for a messenger that an epithelial cell might make? What? Hormones. Okay. Hormones from glands. Remember, glands are epithelial tissues. And the way the glands work is they make hormones that then circulate around in your body through your blood and cause things to happen. Polarity I've already mentioned, but basically it just means like there's a North Pole and a South Pole. That means there's an apical side, which is the open surface to whatever it is there, and sometimes that has cilia and microvilli. And then there's the basolateral, or the bottom surfaces, which are always attached to the basement membrane, okay, or basal lamina, means the same thing. What is it that holds these guys together? All these, if you're going to have something that's going to form a barrier, you know all of those cells have got to hold to each other really pretty tightly to create that those connections that hold them together. The intercellular connections between epithelial cells are very complex. Now, when I read through that section in the book, I got bored to death because I thought they just went into incredible detail on what makes these cells hold together. It talks at length in your book, I think like 2 pages or 2.5 pages, about tight junctions. Tight junctions is just what holds them together tightly. And I think that's about all you need to know, is that epithelial cells are held together by tight junctions. Okay? I would just leave it at that. And you can kind of skip over about a page or so in there. I don't want to graze it over, but I mean just glance it over, but that's it. Epithelial cells are tightly attached to the basement membrane on the bottom, and remember that epithelial cells do replace themselves, they can repair themselves, and so forth. Let's talk about glands for just a minute. Remember, glands are epithelial tissues. And there's 2 basic kinds of glands, and you need to know this, and you'll come back to this a zillion times in A&P. There are endocrine glands and there are exocrine glands. Okay? Now, an endocrine gland, an example of an endocrine gland would be your pituitary or your thyroid gland. Okay? And what they do is they're epithelial cells, but they're all glommed together as a whole bunch of them, and they make these things called hormones, which are the messengers that go around your body and tell your body how to act. And they don't go through a tube or anything. They don't get secreted through a pipe to dump into something. They just are simply released out into the interstitial fluid, the water and fluids that surround the outside of every cell in your body. They have a little bit of fluid around them. And the hormones are just secreted out into that, and from there they are picked up in the bloodstream by little capillaries that go by. The point being, there are no ducts. The other main kind of glands is called exocrine glands, and exocrine glands are things that produce secretions. they secrete out onto an epithelial surface, and they do it through a duct. Anybody here know how to gleek? You stick your tongue out and move away. It was a big deal in middle school. You know, you like, out comes a little shot of saliva, right? And if you knew how to do it really good, you kind of walk up to your neighbor, out would come a few drops. You know, it's kind of a disgusting thought, but that's what middle school is all about, right? Well, we have a whole bunch of exocrine glands in our body. For example, our salivary glands, our parotid glands, they make fluid, they make spit, basically. The fluid is manufactured, it's secreted into the ducts, and there's a couple of openings inside your mouth where the saliva comes out, including the one right there on the end, just below the frenulum of your tongue that makes you gleek, okay? We also have the pancreas secretes a lot of fluid into a duct that then empties into your intestine. The liver makes a whole bunch of stuff called bile that is secreted down through a bunch of bile ducts that then empties into your intestines to mix with your food to help you digest it, and so forth. So the point being is that endocrine glands do not have any ducts, and exocrine glands do have ducts, okay? If you have a duct, then that duct has got to empty into something. We'll learn about it. Let's talk for just a minute about the way in which epithelial cells secrete. And there's three basic ways that epithelial cells secrete. And you need to know these. First one is called merocrine. Merocrine secretion looks like this. An example of it would be a salivary gland in your cheeks here. And you have, this is the superficial layer of the cells. There's a nucleus down here. There's a bunch of mitochondria around in here. You've got a Golgi apparatus that's up in here. And that Golgi apparatus is packaging up these fluids, this spit, saliva. And then the little vesicles, secretory vesicles, move up to the surface, and then they kind of open out. What do we call that process of opening out? What? Exocytosis, right. Exocytosis. Remember, there's endocytosis, where we take stuff in. There's exocytosis, where we... So, merocrine secretion, the first type we're talking about here, involves exocytosis of secretory vesicles. Notice that the cell stays pretty much the same. Forms all these secretory vesicles, they move up to the surface, they get released, bingo. The second type of secretion is called apocrine. The way apocrine secretion works is like this. A bunch of secretions is made. Example is the mammary gland, breast. And so the Golgi apparatus creates all these packets of these wonderful proteins and fat and so forth that your baby's going to live on. And it puts them in these packets. And then all those packets move up here to the surface, but they don't all go to the surface when each open up. Basically what happens then is that the cell throws off half of it. The half that had all of the vesicles in just gets extruded. And this thing then breaks down, and that becomes the milk, okay, in a mammary gland. And so, and then what happens is that part of the cell regrows, it fills up with little packets again, and then it gets cut off and thrown off again. That's called apocrine type of secretion. Notice that the cell is still there, but half of it gets thrown off in apocrine type of secretion. The last type, which you probably have figured out by now, is called holocrine. Holocrine. When I say holocrine, what do you think is going to happen? The whole cell is going to get thrown off. That's right. So in holocrine secretion, and a place to see holocrine secretion is in the sebaceous glands. This is a hair follicle. This is a little muscle that's attached to your hair follicles. But every hair follicle on your body has its own little sebaceous gland. That's why you comb your hair, and that's the oil that forms in your hair. That's why you women spend tens of thousands of dollars every lifetime on shampoo and crap like that. It's because you've got these sebaceous glands that make oil, right? So you've got to get rid of it, I guess. Holocrine secretions, the cells are created down here at the bottom. You see there's a bunch of mitosis going on because a lot of new cells have to be made all the time. They gradually work their way up towards the surface. They fill up with all the secretions, and then the whole cell is blown up and it dies, and that all becomes part of the secretion. Now, has anybody in this room ever had a zit? Yeah, right? I had thousands of them. I was the acne star of my class, I think. But anyway, this is what acne is when one of these little glands right here, and they're very small, attached to every hair follicle, if they get clogged up right in here, see that's the duct. This is an exocrine gland, right? Not an endocrine gland because it has a duct. So it secretes this oil, and this oil moves its way out that little duct. And if that little duct right there gets clogged up with a piece of dirt or a piece of makeup or any of the stuff you guys smear on your faces, right, then it will plug it up. And when it plugs it up, it doesn't send a message and says, hey, we're plugged up. Y'all can stop making sebum now. The name of this oily secretion that comes out. But it keeps making it so that this thing here just starts swelling up and fills up more and more and more with sebum, which is the oil that's supposed to be on your hair, but because this little opening is plugged up, it can't, until it becomes a bump that sticks up out here, and then if it gets infected, it gets red, and then you have to go to the mirror, and you have to go, out it comes. So it exits that way instead of exiting the way it's really supposed to. Okay? That's a zit. That's a hair follicle. That's a sebaceous gland, sebaceous, which is a holocrine form of secretion. So you can kind of bet being holocrine, it's going to be really the thickest of all the secretions, right? And it is. As long as we're talking about secretions, let's talk about three. You know, this is the trouble with AMP. Some guy feels like he has to split everything up. We've got splitters and lumpers. I think I've already talked. Have I mentioned that to you already? Well, this is a splitter thing, okay? They figured, well, we can't just have clear fluids and cloudy fluids, or thin fluids and thick fluids. We've got to give them fancy names, right? So let's call serous glands serum, serous, watery, okay? Serous glands. Mucus glands, thick, cloudy, full of proteins. We call that mucus, right? And you've all had a cold or something and coughed up a whole bunch of mucus from the glands in your bronchial tree. And then guess what? You've got to have something in between, okay? So in between is the mixed endocrine exocrine glands, and they make both serous and mucus, so they're sort of thick, not so thick. How important is that? I don't know. Not very in my mind. All right, let's talk about how these glands are formed. What do they look like under the microscope? Okay? Well, the first one is the easiest, I think, and these are the unicellular glands. In other words, when it says unicellular, what does that mean? One cell. Unicellular, right? You're going to have to start breaking every word we use in ANP. You need to break it into its parts, and then it'll all make sense to you really pretty quickly. All right, so unicellular glands are often called goblet glands or goblet cells, and the reason is because they look like a goblet. I'll show you some examples in a second. They are the only unicellular exocrine gland. Okay? In other words, they do drain out onto a surface. It's one cell that dumps it out onto your respiratory epithelium, for example. And usually what you do is you see all this epithelial tissue, and then every so often there's a single little goblet cell that's making the mucus, it's putting the mucus out onto the surface. Same thing in your intestine, on the inner lining of your intestine, you have a lot of goblet cells. Now, we would like to have mucus formed on the inside of our intestine because we've got all this crap, so to speak, that's got to move through us, right? And when it moves through, we like it to slide on through without any trouble. And so to do that, we want the surface of our intestines on the inside to be covered with mucus. Make sense? Well, so we're going to see a lot of these mucus glands and mucus cells, goblet cells, as we go through this stuff here. First of all, let's talk about multicellular glands, because most glands are multicellular. The only single unicellular one is the goblet cells. Multicellular glands, they can be classified in a bunch of ways. I think this is really silly. Please do not learn this stuff. Do not learn the difference between simple or compound, tubular or alveolar, or branched. It doesn't matter. We're not going to ask you which glands are which, or that stuff. So I think that the order of this is kind of silly. Here's the picture that describes all of that. Here's a simple tubular. That makes sense, right? How about a simple coiled? How about a simple branched tubular? How about a simple alveolar? See, it kind of comes down here to a, what do you call those things at the end of the streets? A little cul-de-sac, yeah, a little alveolus, a little cul-de-sac. And here's a simple branched cul-de-sac alveolar type gland, okay? You do not have to know which type of glands are which kind of these. I just think that's a silly descriptive term. Okay, so that's kind of the glandular end of things. Remember I said epithelium over here. There's all these epithelial types. That's what we're going to start into now. But the other weird kind of epithelial tissue is glands. And so we just talked about them. Talked about exocrine glands with ducts, endocrine glands without ducts. Talked about 3 kinds of secretion: merocrine, apocrine, holocrine. Okay? All right. How do we name epithelia? So we're going into the main types of epithelia now. And let's just start by saying they're easy. It's really easy to understand the naming of them. So if we had a piece of epithelium over here, here's the basal lamina, and above the basal lamina, there was one layer of cells, like that. We would call that a simple epithelia. Simple epithelia simply means, get it? Simple epithelia means one layer. And there's only one layer of cells. On the other hand, if there's more than one layer, so if it's like this, and there's another layer, or maybe there's 2, maybe there's 3, maybe there's 25, it's called stratified. Stratified rock, different stratifications of most everything. Okay, so epithelia can be either simple or it can be stratified. Easy enough. The second word is based on the shape of the top layer of cells. The important word here is top. Top layer of cells. Okay? And they can either be flat, in which case we call it squamous epithelium, flat cells. It can be cuboidal, cubes, cuboidal, square, or it can be columnar, in which it's usually kind of tall, like this. Cells are quite tall. Okay? Now remember that it's based on the shape of the top layer of cells. So a lot of times, epithelium might look, I'm going to erase this, it might look like this, and you know, those are obviously cuboidal cells down there, but by the time they get up here to the top, the cells get thinned out until they're really flat and they're squamous. Okay, we name them by the type of cells on the top layer. Okay, that's the point. Okay. So, just so you know, we're going to have simple squamous, simple cuboidal, simple columnar. We're going to have stratified squamous, stratified cuboidal, and stratified columnar. Okay? 6 different types. I'm going to go over them one by one here in a minute. Now, we also have 2 others. And believe me, you better know these because they have a way of always showing up on tests. One is called transitional. And these are kind of oddballs. Transitional epithelium is really only found in one place, and that's on the inside of your urinary tract, of your bladder and your ureters. What does that mean? What do you mean transitional? Think about it. Why would you need your bladder to kind of have a different kind of epithelium? So it can expand and get smaller, right? Because your bladder goes from this tiny little thing that's completely shriveled up and generally has less than 2 cc's of fluid in it, and then you sit here through this long class and you've had all this coffee to drink this morning and you're making all this urine, and by the end of the hour your bladder is nice and big and it's pushing on other things and you're kind of getting the feeling like, I gotta go. Okay, well that's transitional, and that's the body's adaptation to that makes it able to do that. It makes this epithelium that can get really stretched and then it can come back to normal and can do that over and over throughout your lifetime. The last one is called pseudo-stratified. And I'll go over pseudo-stratified when we get to it, but let me just have you break that down. Pseudo means fake. It's fake-o. It's not really. It's not stratified. But it looks like it's stratified. It's pseudo-stratified, okay? So I'll show you examples of that in a few minutes. That make sense? So that's all the different types of epithelia that you're going to need to be able to identify under the microscope, and you're going to need to be able to give me an example of where each of these is found in the body, okay? Here's what simple epitheliums look like, going back over what I just talked to you about. Basically, a simple means there's just one layer, period. This is stratified. Here's the basal lamina. There's a whole bunch of layers. And remember, they're always going to be named by the shape of the cells on the top layer. Squamous, flat, cuboidal, about square, columnar, tall, forming columns. Simple squamous epithelium is one layer thick, right? And all the cells are flattened out and thin. Where might you find simple squamous epithelium? In your capillaries, on the inside lining of all your blood vessels, that's right. The inside lining of your heart, the inside lining of all your capillaries. Why would you think in your capillaries you would want them to be lined with simple squamous epithelium? I'm sorry, speak up. So it's easy for things to pass in and out. That's right. Because, you know, when your capillaries go through your lungs, they've got to absorb all this oxygen, and they've got to get rid of all this carbon dioxide. You know, in goes the good gas, out goes the bad gas, all that stuff. And it's got to go through these thin layers of epithelium to get in or out. So it makes sense that in places like that, you would have simple squamous epithelium. We also have simple squamous epithelium on the inside of our mouth. Okay, and so maybe when you guys were back in 112, you did a scraping of the inside of your mouth and smeared them out on a microscope slide and looked at them. Simple squamous. Okay? Here's an example of simple squamous looking at it from the top. It sort of looks like that schematically, like this. You can see the nuclei nicely. You can see the tight junctions between the different epithelial cells. Another place where we see simple squamous epithelium, and you all ought to look at this in the lab, is on the inner lining of this little cavity right here. This is called a Bowman's capsule. This is inside your kidney. The Bowman's capsule is the thing that surrounds a glomerulus, which is nothing more than a glom of little arteries and veins. And basically, as the blood flows through there, it pushes this fluid out of this little vascular network here, pushes it out into here, into the capsule, and then it works its way on downstream, and it's eventually going to become urine. Okay, we'll learn about how our body makes urine and how it processes it and all that stuff. But just know that the inner lining of the Bowman's capsule, which is seen here, is simple squamous epithelium. It's really a nice example of it, by the way, and you'll want to look at that in the lab. How about these cells out here? Do you see them? They don't look like simple squamous, do they? Yeah. Those are actually simple cuboidal cells that are going around a little duct right here. There's an opening right there. We'll talk about that later. Here's another example of simple squamous epithelium that somebody already mentioned on the inside of capillaries. This is a blood vessel from here to there. It's a blood vessel. These are blood cells that are laying around in there. And remember, we don't look at things that are still alive under the microscope. They're all dead and they've been frozen and cut up and stained and all that jazz. And so the blood vessels, the blood cells tend to always go to the bottom, whether that's the top or the bottom. So these are all the blood cells. This is the serum, the clear fluid in your blood, and then along the outside here is a lining of simple squamous epithelium. We also call that endothelium. Endothelium is inside of the blood vessels. Okay, now why would we have simple squamous? Well, you've already mentioned it, sort of. One of the reasons is because simple squamous is very important in diffusion. It allows things to pass through it very easily. So the alveoli and the respiratory system, that's a good place to have simple squamous. I mean, your lung, go look at lung tissue, and all the alveoli, the little air pockets, the little cul-de-sacs where the air comes down to every time you take in a deep breath, they're aligned with simple squamous. Simple squamous also plays a role in filtration. Just like in those capillaries we talked about, we need to filter things through the walls of our blood vessels. And so capillaries, the lining of capillaries can allow fluid and other substances to go out, but yet it holds the blood cells in, holds the proteins inside that we need to stay in our blood. Lubrication, we make lots of lubricating substances. We talked about the peritoneum in the belly, for example, that body cavity, how it's always got a little bit of fluid in it that allows our guts to kind of roll around there and move around and not creak and hurt, but they're free to move about. And so that needs to be lubricated, so that's another function. That was simple squamous epithelium. Now let's talk about simple cuboidal epithelium. A simple cuboidal is same as, it's simple, so it's only one layer thick, one cell layer thick, and the cells look like cubes. They're about as wide as they are tall. They have big nuclei in them. Usually the nuclei are sort of towards the middle, and they do lots of things for us. And here's a nice example. This is that kidney again, not the Bowman's capsule, but out in the rest of the kidney. And you notice that these are tubes that run through the kidney that are carrying urine. And the fluid is running through here, and the tubes are all, like any tube, it's lined with epithelium, right? And indeed, it's one layer thick. 1, 2, 3, 4, it's all just one cell layer thick in each tube. This is a separate one. So this is the basement membrane going all the way around it. Here's another one, basement membrane going all the way around it. All of these are the same thing. Okay, and notice they are cuboidal. They're not thin and flat. Why might that be important? Well, that's a good point. Yeah, there's a certain amount of that. There's a lot of stuff you don't want to put back into your body, but there's also a bunch of stuff you want to put out into your urine. So these cuboidal cells are not only being really careful with what gets into the body out of the urine that you started to make, and it puts other stuff out into the urine that it wants to get rid of. So cuboidal cells are thicker. They are packaging stuff up to go back into the urine, and they're pulling stuff out both. Here's a beautiful view. Wow, isn't that pretty? You got to look at the beauty in this sometimes. I'm telling you, it's important. All right, so this is a duct in the kidney. Nice basement membrane going all the way around it right here. Nice, simple cuboidal epithelium in it right there. Notice that this is one tubule. Here's another one over here. Notice that in between the tubules, there are little tiny blood vessels and other little cells that are in between there that are connective tissue to help fill out the differences. All right, so what are the functions of simple cuboidal? Well, as I just said, you can use them for secreting things. And the thyroid, which is an epithelium, remember it's a gland, it has beautiful simple cuboidal epithelium. I'll show you some examples of that here pretty quickly, I believe. And in the kidney, for example, the cuboidal cells tend to absorb things and process which ones they want and leave what they don't. Columnar, simple columnar. Looks like this. They're tall. The oval nuclei are usually towards the base, towards the lower end. Goblet cells are often found in this layer, and the surfaces may have microvilli or cilia. So we might call this stuff ciliated columnar epithelium. It has cilia on it. Here's an example of simple columnar epithelium. This is a duct in your parotid gland. Your parotid gland is this big spit gland in your face right here. John McCain, who ran for president a couple of years, 4 years ago or 8 years ago, he's had a tumor of his parotid gland, so he's kind of got a dimple that goes in right there. And you may have seen people that have had parotid gland surgery before. It tends to form tumors once in a while. So here's simple columnar epithelium. This is one place you can find it, is in the parotid gland. Here's simple columnar epithelium. This is in the intestine. Now the intestine has villi as well as microvilli. So these are actually big villi sticking up into the surface of the intestine. They go up like that, and up like that, and up like that. These are blood vessels. This is one big villi coming up and back. Notice that along the edges, the surface, the epithelium is nothing but simple columnar epithelium here. In the middle is all the blood vessels and so forth. So this epithelium is going to absorb your food. Going to absorb stuff out of the intestine, the lumen of it right out here, into the system. Then it's going to process it and put it into these blood vessels that are running down the middle of the villi, there and here, and send it on to your liver to get processed and nourish you. Please notice that in this epithelium, every so often there's one of these nice kind of roundish looking cells. There's one maybe, here's one. Those are goblet cells. Those are those unicellular exocrine glands that secrete mucus out onto the surface to lubricate your intestine. We have beautiful examples of these over in the lab in the boxes. So simple columnar, their functions are mainly secretion and absorption. For example, in the stomach, they make a whole bunch of enzymes. They make hydrochloric acid. They make the enzymes that start digesting your food. And columnar epithelium also absorbs, so it processes the food and brings it into you. Okay, here's that weird one we talked about, that pseudostratified columnar epithelium. Okay, Pseudo-stratified means it's not really stratified, it looks like it is. And if you look at this picture right here, you notice that there are nuclei, and the best way to identify this under your microscope is to notice that there are generally 2 layers of nuclei. Some are up and some are down, kind of like that. If this was simple, they would all be in one nice row, okay? But the point being here that every one of these cells goes all the way from the bottom and all the way to the top, okay? So it looks like it's stratified. It looks like there's 2 layers, but there's not. They all go from the bottom, from the basement membrane to the surface. It's pseudo-stratified. It says here some do not reach the free surface. Okay, I thought they all went to the surface. But anyway, nuclei are seen at different levels. That's your best key to identifying this type of tissue on a practical exam and under the microscope. They may be ciliated. If it's ciliated, we then call it PCCE, pseudostratified columnar ciliated epithelium, PCCE. Here's an example of PCCE. Notice that the nuclei are at different levels here. Notice that there's a few goblet cells. I think that's a good goblet cell right there. And if you could see this better, you would see that these are cilia up here on top. Here's a much better picture of it. Here's the basement membrane down here below the epithelium, right here. Notice it looks like there's kind of a row of nuclei down here and a row up here. Notice that they're ciliated at the top. So therefore, we call this PCCE. This is pretty much found in the respiratory tree. We'll talk about it a lot as we go through the respiratory chapter. Lines the upper respiratory tract, sweeps debris up towards the throat, may contain goblet cells that secrete mucus. Just for a piece of information for those of you, I'm hoping there's nobody in this room that smokes, but if you do, here's why you should stop. These cilia are very important in sweeping debris up towards your throat. And without those cilia working like crazy to push anything that got through your nasal hairs and through your nose, got all the way down into your respiratory tree, you want these cilia to work to push all that stuff up so you can hock it up and swallow it down, right? Get it out of your lungs. Keep your lungs clean. If you smoke cigarettes, every time you smoke a cigarette, it paralyzes all of these little cilia for about 12 hours. Now, most people I know that smoke don't smoke less than 2 cigarettes a day. They usually smoke, half a pack or 2 packs or 3 packs or 5 packs a day. So basically, their cilia never work. They're always paralyzed. And if your cilia are always paralyzed, that means every time you smoke in that crap, not only the stuff in the cigarette, but also all the stuff that's in the air we all breathe anyway, it doesn't have a good way to get out. So for the rest of your life, it just sort of sits there, and you cough a lot, you bring it up. But this is basically the cause of COPD and emphysema, is this junk that came down into your breathing tubes, and you couldn't get out because your cilia were all paralyzed. That's really where cigarette smoking kills people. Yeah, it causes cancer once in a while, but it kills people mainly by destroying their lungs, and which then destroys their hearts and gives them bad heart disease and so forth. So smoking is just a terrible habit, and I hope after you finish A&P you'll have a better respect for that. Yes?
Does secondhand smoking do the same thing?
Absolutely. Yeah. Let's talk about some stratified types of epithelium now. These have more than one layer. The stratified squamous epithelium is the one that you'll probably see the most of. It's the stratified type of epithelium you'll see the most of because it's got a bunch of layers of cells up top. Remember, we name it by the top layer, so that's the layer that's flat. The bottom layers can be any shape, although they're usually cuboidal. And it functions to protect us in the underlying areas that are subject to abrasion. So our skin is the best example of stratified squamous epithelium. The skin usually has about 40 or 50 layers of cells in the epithelium. Most of them, as they get towards the top of the surface, they're dead. That's why your skin is dry. They also get filled up with this stuff called keratin, which is what makes it kind of dry and leathery and tight. But basically, that's the function of stratified squamous epithelium. Now, it does occur in other places. This is the inside of your esophagus. This was the old Greek way of spelling it, the oesophagus. So this is the lining of your esophagus. So this is where you swallow it, and your food comes down. You notice that this is the basement membrane down here. There's a whole bunch of layers of cells coming all the way up here. But it's called stratified squamous because the cells on top are flat. Okay? Bottom layers can be any shape. Down here they look pretty much cuboidal. This is skin. Well, non-keratinized skin. So this can be inside your mouth or inside your anus, inside your vagina. Any of the tubes that lead to the outside of your body, when they go from the inside to the outside, they are generally stratified squamous, non-keratinized. Keratinized means leathery. So this type of tissue is always moist on the outside. It's inside of us. Once again, here's the basement membrane down here at the bottom. Cells start off pretty fat and almost columnar, working their way up. By the time they get to the top, they're very flat and stratified squamous. This is keratinized, which is what skin is. It's really the only place that we find this type of tissue. Keratinized means that the cells on the outside are cornified or keratinized. Keratin is this protein that is laid down in the cells as they work their way from where they're formed down here. As they mature, they come up to the surface. It takes about 15 days for the cells to turn over from going to the bottom to the top up here. They're all full of keratin, which makes them leathery. So whenever you see a slide of skin, it will always have this corny looking stuff. It looks like it can flake off. We won't try to fake you out and not show you this. That's what makes it skin. It's this flaky layer on top. Now, how many of you use a bathtub? Most people don't anymore. We usually use showers. But if you take a shower, if you take a bath, and you get out and you soak in the bathtub for half an hour, and you soap up, and you do all that wonderful stuff, and then you rinse off in that filthy, dirty water, and you let the water out, you know there's this little ring around the tub, right? Now, what do you think that ring around the tub is? It's dead skin. It's that cornified stuff from the top up here that's flaking off all the time. And you can scratch your skin enough and you'll see it kind of flaking off. So we're losing our skin all the time. It's a constant process of replacing our skin and it working its way to the top. So it's only found on the epidermis of the skin. Keratin is this protein that helps strengthen the cells against abrasion. You wouldn't want, you want to be pretty well protected on the outside of your body out there against abrasion. We'll talk about transitional and then quit. Transitional epithelium is the one I said is the one that has to transition, like on your bladder, to stretch and not stretch. If you learn one thing about transitional epithelium, let me tell you, the thing you want to know is that the apical cells, the cells on the top, are dome-shaped. Well, what does that mean? Well, that means that these cells that are sticking up there on the surface are dome-shaped. So, you go up here to the top, you will see some cells that kind of stick up there like that. They're big dome-shaped cells. And you might figure that because these are the cells that are going to have to stretch, right, and go from being dome stretched to being stretched out flat. So when you see the word dome shape, you know we're talking about transitional epithelium. And this is only found in the urinary system. That's the only place you'll find transitional epithelium. You all will need to look at that under the microscope. It's basically, its function is simply to allow the expansion and recoil. So here's a picture of it right here. This is a distended bladder. This is all stretched out right here. And notice the cells on the top are all stretched out. And they're pretty flat. But when you pee and your bladder empties and the wall gets thick again, the cells on top can look like these guys. And they're dome-shaped. That's what that means. Okay? Now, let me tell you, this is a really nice picture. This is kind of a classic picture. Because what you'll see when you look in the lab, in the boxes that say transitional epithelium, is you'll see one of these about every 20th cell. There's not, you have to look for them. Okay? All right, we'll stop. We'll finish on these last few stratified ones later. They're pretty simple. All right, in lab today we're going to do the rat, and I think you'll enjoy it. How'd that go? Good. Yeah. Unrelated question, okay?
So, at my work, I have somebody who is on hospice, and they've been giving her morphine and lorazepam.
Yeah.
How does morphine?
Okay, this is our second lecture on the tissue chapter, chapter 4. And except it's not projecting on the screen, and that's probably because I don't know what I'm doing. Let's see. What I need to do is close that thing down and close that thing down, and then probably just yay. Okay, this very unimpressive slide here is an example of stratified cuboidal, and these are little ducts here, and they're lined with 1, 2, 1, 2, 1, 2, 1, 2. 2 layers of cells. That's enough to make it called stratified, but yet that's all it is just 2 layers thick. So these are cuboidal. I think that's pretty obvious. And that's all you got to know there, really. Here's another one. This is a sweat gland. And once again, 1, 2, 1, 2, 1, 2, 1, 2, so forth. It's just never thicker than just 2 cell layers thick. How about stratified cuboidal? Stratified columnar. It's also very limited. This is a good point right here. Read this sentence right here. It says, to tell the difference between this type of epithelium and pseudostratified columnar. Because remember, pseudostratified columnar, has the nuclei at different levels, even though it's just really 1 cell layer thick. Look at the nuclei. In this stuff here, in stratified columnar, the nuclei are lined up in a straight row, as opposed to pseudostratified, where it's not a straight row. They're found in different areas like the pharynx and the male urethra, lining some glandular ducts in the anus. Here you go. Perfect example. 1, 2, 1, 2, 1, 2. Notice the nuclei are in a nice straight line. The top layer's in a nice straight line. And there's some kind of funny cells in there too. What are those called? Yeah. Yeah, these are goblet cells. See this one here, this one there. Those are those unicellular exocrine glands. They make mucus, keep the surface moist. Another example here, stratified columnar, 1, 2, 1, 2. Once again, 2 lines of nuclei nice and straight. Okay. Now, when we talk about tissues, we're going to talk about the 4 main kinds of tissues, right? And we've now talked about the first one, which is epithelium. epithelium is great because not only does it line everything, but it covers the outside of everything too. So you always see epithelium practically whenever you look at any microscope slide. There's going to be some epithelium. If nothing else, there's going to be little bitty blood vessels running through it because every cell in your body has to have a blood vessel running within a half a millimeter or so of it in order to get nutrition and get oxygen and get rid of its waste products and so forth. So you've always kind of got some blood vessels in there, little tiny capillaries. But now we're going to type 2. And when we get to the practical exam, I will say, what major type of tissue is this? Okay? And when I ask that question, what major type is it? In fact, a lot of times I just say the big 4, okay? Then you'll know that what I'm asking you is to tell me whether it is epithelium, whether it is connective, whether it is muscle, or whether it's nerve, okay? Because that's the big 4 right there. So if you can identify every structure or every tissue as one of those big 4, got about a third of the questions right there, okay? So keep that in mind. Those have been all the epitheliums, and now we're going to the connective tissues. Now, what are the connective tissues? Well, they connect everything together. That seems simple enough. But they also hold us together. They provide structure, for example, bone, and the other big one there is cartilage. Bone and cartilage are 2 connective tissues. They store energy. I've stored up enough for 2 winters. Fat, okay? So we all have some of it, but fat sort of fills in the spaces between everything, and then we add to that. Our blood is a connective tissue. Now, that may seem a little odd, blood as a connective tissue, but think about it. It fits in between everything. It transports things around. And one true point you can always say is that you can never see connective tissue. When I look at you, I can't see any connective tissue. Even if I look in your mouth or anywhere, any of your orifices, I can't see connective tissue. All I can see is epinephrine. Is epitheliums from the outside. Okay? So just kind of keep that in mind. And if you know it's not on the, well, that's what a connective tissue is. This is the main kind of connective tissue that you need to look at, and you'll need to look at this under the microscope. You remember the other day when you were in lab and I said cut the crinkly stuff? When you're separating the muscle layers and you're separating things inside and dissecting things apart and you're cutting that little whitish kind of crinkly stuff. Well, that's what this is. That's called loose areolar tissue or loose connective tissue. It's also called connective tissue proper. 3 names for the same thing. Okay? And what it is just a loose kind of stuff. There's a lot of just water in it, a base material, and then scattered in it are all these different cells. Now, the first 2 things there's always going to be is there's going to be fibers, and there's two major kinds of fibers. One is elastic fibers. Elastic fibers are always really thin, usually black on our standard microscope stains. And then the second one is collagen fibers. Collagen fibers are the big wide pink ones. Okay? Now, as you might guess, elastic fibers give you elasticity. So when I pinch my skin up and make a little tint out of it, it snaps right back down and gets smooth again. Until I get to be about 85, and then you probably pinch it, it'll stay up for 5 minutes, you know. But that's just the way we age. We lose our elasticity in our skin. So that's the 2 major types. We have one other kind of fibers we'll get to in a little bit. So elastic, collagen, and then there's a whole bunch of cells scattered around through this tissue. Probably the most common cell in there is a fibroblast. What do you think fibroblasts do? Fibroblasts. blast. They what? No. Well, they do secrete protein. They make the fibers, okay? Fibroblasts make fibers. And that's one of those little syllables that you need to know about. You need to know that blast, and there's all kinds of blasts. There's hemoblasts and hemocytoblasts and chondroblasts make cartilage. There's osteoblasts that make bone. Anything that has a blast on the end of it usually is the cell that makes that tissue. Okay? So just file that away in the back of your head because we're going to come back to that a whole bunch of times. Fibroblasts are illustrated here. There's also several other kind of cells that we'll get to on other slides, but there's macrophages. What's a macrophage? Excuse me? No, not really white blood cell. Yeah, they eat stuff. They clean up garbage and eat bacteria and eat dead cells and all that kind of stuff. They're basically the SERVPRO system in our body, like it never even happened. Now here's another diagram of the same thing. This is a schematic diagram, a little more, has a few more in here. Here's the fibroblast they've drawn like that. There's a free macrophage. This is a mast cell. You need to know something about mast cells. Mast cells are the ones that that release histamine. They also release heparin, which is blood thinner. So that's mast cells. There are both fixed macrophages and free macrophages. You can't tell the difference looking at them under the microscope. And don't feel like you're going to probably have to identify many of these cells under the microscope in this kind of tissue. But anyway, here's a melanocyte. Melanocytes make melanin. Melanin is what makes our skin darker colored. So depending on our race, we have more or less melanocytes. And actually, I should say that we have more or less packets of melanin inside the melanocytes. That's really the difference between the darker races and the lighter races, is how much melanin you have stored up in your melanocytes. When I think about all the problems that the world has had and we still continue to have with race, and I look at the difference between the races under the microscope, it's incredibly small. It's like one layer of cells in the deepest layer of your skin that is, is the whole difference. Yes?
I was just going to say, isn't it more like your ethnic makeup as opposed to race, which is like a socially constructed term?
Well, maybe that's a good point. I'm not a social scientist, but good point. And we can make ourselves darker, right? We can go sit under the suntan light or sit out in the sun, smear all that crap over our bodies, and we can turn darker. Or we can be like Michael Jackson and smear all that stuff on our bodies and make ourselves lighter, right? We'll get to that when we get to skin next chapter. Okay, plasmacytes make plasma cells. Here's a little tiny blood vessel running through here, and it has a few red blood cells in it. Mesenchymal cells. When we say the word mesenchymal, that's another word you ought to kind of start to think about. Mesenchyme means, let's just say if I said mesenchyme means stem cells. What does that make you think?
Yeah, where we came from, like the original cell.
Right. Mesenchyme, mesenchymal cells, and stem cells are just kind of immature cells that can still change into different things. So a mesenchymal cell can turn into a fibroblast, or it can turn into a macrophage. It can turn into different things. Okay. Adipocytes, adipose tissue is fat, and adipose cells look like that. I will tell you that most of the time under the microscope, the fat, the little globule of fat that makes up most of a fat cell is actually gone. because the fat dissolves when they put the staining stuff on it, and that just washes it away. So it just looks empty instead of looking like fat, like in that picture right there. Yeah. Oh, I'm sorry. Okay, so let's go through the list of cells that are in the connective tissue proper. Fibroblasts, that's the most common type in loose connective tissue. You can always find it. It makes these proteins. It makes hyaluronin, which is basically the cellular cement that kind of holds everything together. Fibrocytes are the second most abundant cell. And what do? Well, they maintain the fibers. So here you go. Think about this. Blast cells make things, and then cytes take care of them and maintain them. So fibroblasts make the fiber. Fibrocytes take care of the fiber once it's been formed. Okay? And that same thing will go for chondroblasts, chondrocytes, osteoblasts, osteocytes, and so forth. Okay? So just keep breaking these words apart. Fibroblast, fiber cells, fiber makers. Macrophages are these large amoeba-like cells that eat up stuff. There's two kinds that you can't tell the difference between. I think I mentioned that before. Fixed macrophages just stay out in the tissues. Free macrophages can migrate. They can go in and out into the bloodstream and out of the bloodstream, but mostly they're out in the tissues. So if you get a splinter in your finger, the macrophages that are in that area, the fixed ones, start trying to eat up the splinter. But also some free macrophages will come to the area and help fight it. And so, and if they keep coming enough, then you get this whitish, gooey stuff around your splinter, and it's called pus, right? And then you finally get the splinter out, and the pus drains out. Well, that's really just a bunch of macrophages and a bunch of white cells from your blood as well. But that's your body's attempt to fight off that splinter and get rid of it, eat it up. So adipocytes are fat cells. Each cell stores a single large droplet of fat. They're also called signet ring cells. Signet ring cells. And that's because they look like this, they're shaped like this, and kind of shaped like a signet ring, looking at it from the side. So this isn't exactly a signet ring. A signet ring has a flat surface with your initials on it, right? But get the idea? And that's the nucleus over here on this one side that accounts for its shape. Mesenchymal cells, like I said, they can differentiate into different things. They're stem cells basically that can respond to injury or infection and differentiate into other kind of cells that the body might need in that area. Melanocytes synthesize and store the brown pigment melanin. Mast cells are very important in inflammation. They release histamine. You guys have probably taken antihistamines, right, to dry up your nose and stuff. That's because you had a reaction to some virus or some pollen or some antigen, and you released all this histamine, and it's helping your body fight off that, but at the same time it's making you miserable, so oftentimes we take antihistamines to treat the symptoms. Basophils or leukocytes that also contain histamine and heparin. Leuco means white. Yeah, white. Erythrocytes means red. So red blood cells are called erythrocytes. So once again, just keep breaking the words down, and I think they'll make pretty good sense to you. Lymphocytes. These are immune cells that help us fight infections, and they can turn into plasma cells, which make antibodies. That's why if you have the flu one year, you build up these lymphocytes, and you can never get that flu again. Now the problem is the flu continues to evolve and changes every year. So next year there's kind of a different flu, and in the next year there's kind of a different flu. And that's why we tend to take a different flu shot every year. And that's why the scientists that work on all this stuff are constantly looking for mutations and trying to figure out what do we need to put in next year's flu vaccine in order to make more people immune to it. Macrophages are these little bitty, micro means small, huh? That makes sense? Under the microscope, probably. Macrophages are also phagocytic blood cells, and examples of those would be neutrophils and eosinophils. We are going to talk about blood cells in detail when we get to the blood chapter. That's in 232. But you probably ought to know a little bit about what blood cells look like, and you will from what we do here. Collagen fibers are those big pink fibers. They're the ones that make us tough and strong. One thing about collagen fibers is they only resist force in one direction. So in some types of connective tissue, all the connective tissue, all the collagen fibers are running in the same direction. Perfect example of that would be a tendon. In a tendon, where we have a tendon here on the back of our heel, the Achilles tendon, and it's connecting these muscles up here in our calf down to our heel bone. And all of those, when you look at those under the microscope, you'll see that all the collagen fibers are going in the same direction because it only resists force in one direction. And here's the third kind of fiber that I told you there was collagen, and I told you there was elastic. And here's the last one. It's called reticular fibers. Now, let me just tell you that reticular fibers are hard. I think they're hard for you students to grasp. They provide significant structure. They're pretty damn weak. They're not strong like collagen or even elastic fibers. And they're organized in a reticular fashion so that they're all kind of meshed in amongst each other. And the place you would find reticular fibers the most is in different tissues, which we call reticular tissues. A perfect example of a reticular tissue would be the liver. Now, you all saw the liver yesterday in the lab, and you probably, I don't know if you did this or not, but if you take a little piece of liver, you know, it kind of stands up and holds its shape, but yet if you took and pinched it, it would just all go away, right? So that's what reticular fibers are, and they support things like the liver. They hold the spleen, so the spleen maintains its shape. They are in lymph nodes, another example of a really kind of soft tissue, but yet it does have structure and formation. Elastic fibers, we mentioned, they contain elastin. That's the protein that gives them their function. The nice thing about elastic fibers is they can stretch and they can come back to normal shape. They're like rubber bands. The book uses this example, and I guess we do in the lab too. There are some elastic ligaments of the vertebrae that go between your vertebrae bones back here, and they allow you to bend and then snap back. And they lengthen and shorten. And that's probably one of the places you find almost pure elastic ligaments. We'll show them to you in lab. Ground substance is the background noise, the background clear, colorless, viscous, kind of thick fluid that fills in the spaces between all of these cells and all of these fibers, and it slows the movement of pathogens. What's a pathogen? Pathogen is something that makes you sick. Pathology. Pathogens would be things like bacteria, viruses, parasites, and so forth, so that they can't invade our connective tissues too quickly. Adipose cells, they are the fat cells. Oh, what a cute baby. He's so chubby. Isn't that cute? Well, let me tell you, you don't want to have a cute, fat baby. Because if you give that baby a whole bunch of cells, a whole bunch of fat cells, that baby will probably struggle with weight its entire life. We make most of our fat cells as infants, and from that point forward, yeah, we can gain weight, and when we do, what we're doing is we're taking that same number of fat cells, we're just filling them up with fat. But we're not making more fat cells, we're just filling the ones we have up more. And so people that don't start off life with too many of them tend to be able to control their weight better throughout their life. So avoid the cute little fat baby. It's not what you want. Put them on a diet when they're about 2 months, if they're too fat. I wish my mother had done that. So, adipose, here's the point here, this is the teaching point. Adipocytes in adults don't really divide. They just fill up the fat. You got the same number, you just got bigger ones, okay? Mesenchymal cells can differentiate into more fat cells, but that's pretty unusual. You got to really be hogging it down for your body to decide it needs to make more fat cells. Excuse me. Did you laugh? That was supposed to be a joke. It's okay. All right, reticular tissue I told you about. It's in things. Oh, I'm sorry.
I just had a question about the fat cells.
Yeah.
If you get liposuction, are you removing those fat cells
Yes.
from your baby?
You're sucking out a bunch of
They
cells.
don't grow back?
You'd like to think that, but the ones around it can certainly make up for it and get bigger and make up for it.
I thought when you were an adult, I thought you could get more fat cells. That's interesting.
Well, you really don't. That's what our book teaches, and I think that's a good point. Okay, so these reticular cells have this complex three-dimensional network. It's a bunch of fibers that are kind of all connected like this. So the reticular organs, I think I mentioned all of these except bone marrow. Spleen, liver, lymph nodes, and bone marrow is another example of a reticular tissue, which means it has a whole bunch of reticular fibers in it. I need to have a picture to show you. Here's some pictures now. This is these are fat cells, signet ring cells. Here's the skin, and see down here in the deeper layers of the skin, you'll see a lot of fat cells in virtually every body. Here's a question for you. What is the chemical that makes up this yellow stuff? You guys just studied the chemistry chapter, right? What is that? Yeah. It's a lipid, yeah. What kind? Yeah. It's a triglyceride, that's right. So fat is triglycerides. And think of that when you're looking at them under the microscope. This is reticular tissue, and this is a piece of liver here. And they've pretty well dissolved away all the cells that are in it. It just looks like there's a bunch of nuclei there. And they've left the reticular fibers, same as over here. Now, here's the point that you need to write down. Reticular fibers can only be seen if they are stained for specifically. Okay? So on every slide over there in that lab, you won't see the reticular fibers unless it says reticular stain or unless it says reticular fibers, because they're just kind of invisible with our normal stains, H and E, hemus, eosin, H and E. Anyway, the pink and blue slides. That's the main stain we use for almost everything. But anyway, you will not see these on any microscope slide unless it was specifically stained for. But on the ones that it's specifically stained for, they're really obvious, okay? So just be sure you've looked at the slides that are reticular slides before the exam. Let's go through some of the connective tissues besides the loose areolar one. Here's dense connective tissue. There can be dense regular, which is like tendons where everything's going in the same direction. They can be dense irregular where it's going in a bunch of different directions. And then there's the elastic tissue, which is all full of just the elastic fibers. Here's a point that you should probably know, and you'll use this many times in A&P. Know that tendons attach muscles to bones, okay? So that Achilles tendon back there attaches my gastrocnemius muscle to the calcaneus bone. All tendons connect muscles to bones. All ligaments connect bones to bones. So you've probably heard of somebody twisting their ankle and tearing their ligaments. Well, that's exactly what they've done. Those ligaments that go from bone to bone down there in your foot get twisted and ripped, and that's a sprained ankle. That's torn ligaments, okay? The way you know it's really a sprain, and you know the way you know they've really torn ligaments, you'll always see blood under the skin. It's got to bleed or it didn't really get ripped, okay? So it'll get black and blue. Aponeuroses, that's a fancy word. Aponeurosis means like a sheet of tough stuff. So like on our abdomen here, we've got these sheets of fascia or aponeuroses that resist pulling in all directions. So that when you do sit-ups or when you're breathing real hard or whatever, you've got this support system out here across your tummy that supports you in all directions. So that's what aponeurosis does. Here's a tendon right here. Notice that pretty much the collagen is all going in one direction. This is showing a picture of the triceps tendon, which is the one that goes from your triceps muscle on the back of your arm here down. Right, the tendon comes a tendon right here, goes across and hooks into your olecranon right there to make your arm go out. Right. Triceps extensions. Anybody in the weight lab? Okay, we're exercising my triceps muscle. Triceps tendon. Dense irregular. We find that in lots of places. Layers of underneath the skin, around the cartilages, around the bones, and it forms capsules on lots of organs. Even the liver has kind of a capsule around it, which is dense irregular. Now let's look at these words here. Periosteum, you've probably heard that word before. Periosteum is the really tough layer that's on the outside of all bones. So like if you go to Kentucky Fried Chicken and you eat a chicken leg and you've kind of eaten all the muscle off of it, and then you take the bone and you kind of break it, there's still this kind of tough stuff holding them together, and if you kind of pull it apart, it kind of strings off of it. That's the periosteum. That's the outer layer of the bone. It's the area that has a lot of, it's dense irregular, it's got a lot of fibers in it, and it's what carries the nerves and the blood vessels to the bone. Okay. Same thing around chondro. Chondro is cartilage. So periosteum is around osteo. Osteo is bone. Perichondrium is around cartilage. Remember we talked about these regions up here being called the hypochondriac. Okay, chondro is cartilage. Same thing. Here's what it looks like. The only difference in this and the tendon is that now the fibers are going in all kinds of different directions. Elastic tissue. The only real example we give you of this is in the spinal vertebrae. I always have a hard time with these slides, but this is where they come from. It's between the vertebrae that hold your head. These are all, they look like collagen fibers, don't they? Yeah, I know, that's the problem. They're elastic. Most of them are elastic fibers there in that picture. Let's talk about the fluid connective tissues like blood and lymph. They're mostly water. They carry a lot of specific cell types, which we call the formed elements. That's a word you ought to know. Formed elements. Well, blood is, you think blood is just water, right? But you're going to learn that it's not. It's a lot of it is water. We call that the serum. But we also have a lot of cells in it. We have red cells, we have white cells, and so forth. Red cells are called erythrocytes. Erythro means red. Erythema. Get a rash or something on your skin and it turns red. We call that erythema of the skin. Erythrocytes, red. White blood cells, leukocytes. And there's also these tiny little things called platelets, which help us clot our blood. We'll learn about later. We also have the fluid part of your blood and the fluid part of your body, really. Plasma is 55% of our blood is plasma. So 45% of it is red cells and a few white cells, but 55% of it is plasma, which is literally just water with a bunch of proteins in it. Okay? Interstitial fluid is the water that is around every cell. Every cell in your body has water around it. Between cells, that's how the oxygen and the sugars and so forth are going to diffuse into all your cells. They're going to diffuse out of the capillaries nearby, and they're going to work their way through the interstitial fluid to get into the cells to nourish them and so forth. Lymph is another fluid. Lymph is kind of extra fluid that our body picks up in the lymph system. It's another set of blood vessels, really, that keeps us from swelling up too much. Here's just a quickie. Here's the red cells. Red cells are called the biconcave disc. I think you learned that in 112, didn't you? It's a kind of a disc-shaped thing, but then it's concave on both sides, so it goes in like a cave. So they're thinner in the middle than they are around the outside. They kind of look like donuts under the microscope a lot, especially if you kind of get them under right at the right power, and then you kind of flick it back and forth. They kind of blink at you. Platelets are really small. They're probably, in relation to red cells, they're probably 1/10 the size they're illustrated right here. Here's a bunch of different white cells, and once again, we'll go through those later. But just so you know, there's eosinophils. Eosin means red as well as erythro. Eosinophils are have these bright orange almost granules. Neutrophils stain neutrally. Basophils stain basically. That's blue. It's a pH thing. Lymph is this extracellular fluid that's fluid outside of the cells, and the lymph is picked up by the lymph vessels and taken back up through the lymphoid system. Basically, all of the lymph in your body, most all of the lymph in your body, collects in these lymph vessels. It comes up through your chest, through what's called the thoracic duct, and then it enters into the venous system, dumps back into your veins right here at the junction of the subclavian vein and the jugular vein coming down from your head. Then it goes back in the veins, and then your body can do whatever it needs to with it. We have a cardiovascular system, which is the blood system we will study in great detail. The cardiovascular system has 3 different types of vessels in it. It has arteries, and the definition of arteries means arteries carry blood away from the heart. Now notice I didn't say that means it's oxygenated. I just said arteries carry blood away from the heart. Veins carry blood back to the heart. And capillaries are where the two connect. So arteries go out, they get smaller, smaller, and smaller, and smaller until they become capillaries. then they start getting bigger and bigger and bigger as they bring the blood back into the big veins, come back to the heart. Lymphatic system I've already mentioned. Let's talk about the supportive tissues, cartilages and bones. We will spend a whole chapter on these things later, on bones and cartilage, when we get to the bone chapter later here in 231. Cartilage is kind of soft and kind of a little bit squishy, so it can act as a shock absorber, and it still can protect us. Bone, on the other hand, is very calcified, and so it's made to support us and hold us together. It also can break. Cartilage hardly ever breaks because it can usually bend. In fact, that little 2-month-old baby we were talking about a few minutes ago really doesn't have any bone yet. It's almost all still cartilage in babies. It gradually becomes ossified and turns into bone as they grow. The matrix of the cartilage is the kind of clear substance that is what really cartilage is. It's made of a substance called chondroitin sulfate. You need to know that. And these are proteoglycans, which means they're proteins, proteoglycans that came from chondroitin sulfate. And these are the proteins that form the ground substance, the background kind of clear stuff in cartilage. Chondroblasts, I mentioned earlier, make cartilage. Chondrocytes maintain the cartilage. They're mature cartilage cells. And so these chondrocytes, which actually live out in the cartilage themselves, are surrounded by cartilage all around them. And they live in a little hole in the cartilage, which is called the lacunae. We'll see that osteocytes do the same thing in bone. Well, lacunae means a little chamber that it can live in and help take care of the cartilage that's right around it. One thing about cartilage is that there isn't any blood vessels in cartilage. And the biggest point I would make about that is if you ever tear a cartilage, for example in your knee, you have a torn cartilage, it's not going to heal. It never heals because it has no blood vessels in it. So if you tear a cartilage in your knee, you're probably looking at going in and having arthroscopy, and they'll go in there with a little shaver and shave it out, make it smooth again, but they can't really put it back together because it never heals. Yes?
So like, if you ever get like your card, like the only way, like the only thing that heals is skin.
In your ear?
Yeah.
Yeah, we'll talk about ears in just a minute. I guess that's my other lesson to make to you about this right here. Your ear is made up of cartilage. It's a different kind. It's called elastic cartilage that makes your ear flip around. And my advice to all of you who seem to like to pierce yourselves a lot is I think piercing is great as long as you put it through your skin. But when you start putting piercings through the cartilage of your ear, just know that you're taking a really significant chance. Because if you get an infection, even a little infection in a piercing that goes through your cartilage, remember the cartilage has no blood vessels, it has no ability to fight infection, and that cartilage, the entire cartilage will turn to pus. And your whole ear will get red and swollen. I've got pictures of one of these that I took care of once upon a time, and it's a horrible problem. Basically, you got to go see a surgeon, and he's going to make an incision the length of your ear and the back here, and just scoop out the pus and scoop out all the cartilage, and you're going to have one very funky-looking ear from that point forward.
Is that like, the UFC fighters that have like the cauliflower ears? Is that why they're all big and
Yes, they've been damaged and torn and broken inside there. Yeah, inside their ear. It didn't get infected because there wasn't a cut through there, but yeah, it changes shape. So there is blood around the cartilages. On the outside of the cartilages, it's called perichondrium, which is that tough layer that we talked about a minute ago that gives you a tough spirous layer and also is what carries the blood and so forth to it to maintain it. Here's an example of cartilage. This is cartilage. All this light blue stuff out here is the cartilage. The cells started off like that. It divided, and as it divided, it started making new matrix, new cartilage out here. It gradually grew apart, and you get more and more cartilage formed. This is what's called interstitial growth. The other way of cartilage growing. In fact, I have actually it's called appositional growth. It layers on. It makes layers on top. Blood. A lot of our bones, as I said before, they start off as cartilage and they get gradually replaced and become bone. I have 2 little videos that I have posted on D2L that you ought to take a look at. Okay, we have 3 different types of cartilage that you need to know about. The most common type in our body is probably hyaline cartilage. We have a big chunk of hyaline cartilage between our thigh bone and our knee bone called our meniscus, and that's why we can run, and it provides a little bit of compression from pounding on the ground. Hyaline is stiff and flexible. It's on the end of every bone, actually, in all synovial joints, rib tips, sternum, trachea. Trachea. Remember those little tracheal rings you saw yesterday in the rat lab on the trachea? Elastic cartilage is supportive. It bends easily. It's really only found in 2 places, your ear and your epiglottis, which is a little flapper valve in your thing here that keeps the food from going down into your windpipe. Epiglottis. So that's 2 places to find elastic cartilage. The third one is called fibrocartilage, and it's a lot like hyaline cartilage, except it's got a whole bunch of fibers running through it. Makes it really tough. Common place is in your pubic symphysis. This is where your 2 hip bones connect in front. And in your intervertebral discs, between each of your vertebrae, you have a little intervertebral disc that tends to give you some shock absorption, allow for a little bit of movement. That's what a slip disc or ruptured disc is. It's rupturing one of these intervertebral discs. Here's pictures of hyaline cartilage. Notice it's nice and clear, kind of light blue usually, or light pink out here. These are chondrocytes that are living in these little lacunae we talked about, the little cavities they live in. Here's an example of elastic cartilage. The biggest difference in elastic cartilage is it just has a whole bunch of elastic fibers running through it. Example would be the ear. And the third one is fibrous cartilage, which has lots of collagen fibers running through it. And as I said, the best example of that is an intervertebral disc here in your back. Bone is very strong. Bone resists shattering. But just know that your bone is not all calcium. About 1/3, I believe, of your bones is collagen. Collagen fibers, those really tough fibers that hold your tendons and all that other stuff together. It also holds your bones together. And that gives us a little bit of flexibility with our bones. We can flex them, and then they eventually will break if we bend them too far. But that's why it resists shattering, because there is so much collagen fiber in bone. Bone cells, osteocytes, are the cells that maintain the bone. Remember, osteoblasts made the bone. Osteocytes take care of it. They live in little central canals around the matrix. We'll talk about the structure of bone quite a bit next chapter. I just want you to kind of hear about it now. The periosteum is that tough layer on the outside of the bone. Just like there's a perichondrium around cartilage, there's a periosteum around bony surfaces. And by the way, when you break that chicken bone we were talking about from Kentucky Fried Chicken, and you can pull it apart, and that kind of stuff kind of pulls apart, which is the periosteum. The only reason you can do that is because that thing has been fried at 400 degrees for 10 minutes or something. You know, that the periosteum has been cooked. Otherwise, it's really tough in a live person. Periosteum is very, very tough, very hard to strip off and so forth. Here's how bone is kind of organized. Bone occurs in these little units called an osteon. An osteon is a unit of bone. It surrounds a little central artery and vein called the central vessels in the central canal. And then these little things right here, this is one, that's an osteocyte, that's an osteocyte, that's an osteocyte. So the osteocytes kind of line up in rings around the central canal. And these, they started as osteoblasts, remember that's what, then that made the bone right around it. And then eventually, as the bone is matured, they become osteocytes. And that the job of this osteocyte right here is to take care of that bone just right around it. Now you might ask yourself, well, how does this bone cell get any oxygen or get rid of its waste or anything like that? And the answer is it does it through these little tiny canaliculi. These are little outpouchings of the cytoplasm. Can I ask y'all to put your cell phones up, please? If you want to play with your cell phones, please do that somewhere else besides in my class. Thank you. These are called canaliculi that run in between the different layers, and they're just little outpouchings of the cytoplasm that allow these cells to get nourished by just diffusion through that. On the outside of the bone, of course, is the periosteum. Tough layer, very tough, fibrous layer and cellular layer on the outside. Let's talk about membranes for a minute. We talked about it yesterday in lab. We talked about the peritoneum. We talked about the pleura. We talked about the visceral layer, which is on the organs, and we talked about the parietal layer, which lines the inside of the cavity. These membranes are nothing but physical. They're physical barriers. It consists of an epithelium. Now, obviously, if it was just an epithelium, it would just fall apart. There wouldn't be anything to it, right? But it has a bunch of connective tissues with it that can kind of give it some substance. When we would open an abdomen, with the last layer we would go through would be the peritoneum, and at the end we would usually always sew up the peritoneum, put it back together. And it was tough enough that you could sew it up and it would hold stitches. In these rats, I'm not sure, that'd be pretty tough. It's pretty flimsy in these guys. Luckily, they don't get operated on much. 4 types of membranes. Mucous membranes are basically the membranes on the inside of you, like inside of your mouth, inside of your anus, inside of your vagina. Serous membranes are the ones that just secrete some water, not much mucus, not very thick. So an example of a serous membrane would be the pleura, or the peritoneum, or the pericardium. The cutaneous membrane is a fancy name for saying skin. Synovial membranes is a fancy name for describing the membranes that line your joints. Okay? Your joints are called synovial cavities. They're filled up with synovial fluid. It's this really greasy, slicky stuff that lives in your joints that allows your bones to slide on each other very easily. I know when we were doing surgery, we would oftentimes be operating on people that had been in a multiple trauma accident, for example, and you'd be operating along, and suddenly your hands just got to where you couldn't hold anything, you couldn't tie anything, you couldn't do anything. They were just so greasy and gross. And then you looked around and you said, oh, geez, they've torn into their hip joint, or they've torn into their knee joint. And this is synovial fluid that's on our fingers. And there was nothing to do but just change gloves and wash it and try to get it washed off as much as possible, and then start back in whatever we were doing. But yeah, synovial fluid is incredibly slickety. Mucous membranes, as I said, they line all the passageways that have external connections. That would be your nose, your mouth, your urethra, your vagina, your anus. A nice way of saying all those things is your digestive, respiratory, urinary, or reproductive tracts. Epithelial surfaces have to be moist. The point where your mucous membrane starts is right on the inside of your lip right there, where it's moist all the time. And you need that not only to be able to absorb stuff into your body through the mucous membranes, but also for it to excrete and make some mucus and make water. Lamina propria is an areolar tissue. It's a areolar connective tissue, which is basically loose areolar connective tissue, like we talked about. Serous membranes they line cavities that aren't open to the outside. They're usually thin but not very strong. They are serous. They just secrete some water. This would be like the pleura, peritoneum, pericardium. They have a visceral and a parietal portion. And the three serous membranes are illustrated there. We talked about them yesterday in lab. Here's what they might look like under the microscope. A mucous membrane is going to have a bunch of goblet cells. Why? Because goblet cells are what makes the mucus. Duh. Serous membranes, like your peritoneum, they just make water, which is just, it doesn't have to be secreted specifically by specific cells. It just leaks out through the surface. The skin is really thick, waterproof, dry. Obviously, it's the thickest of the membranes. The synovial membranes are the ones inside the joint. We'll talk about that more when we get to joints. But it does make synovial fluid, which is this really slick stuff. The skin looks like this. It's a stratified squamous epithelium. Like I said, the deeper layers are not squamous, but by the time it gets up to the surface, it's very thin cells. This would be an example of a synovial membrane in a joint here. The synovial membrane runs along the outside. Most bones have a piece of hyaline cartilage or articular cartilage on the end of them. That is the part that's actually going to do the contact and actually the sliding back and forth. And so that's important. Couple of comments here about the basic framework of how this all gets put together to work. The connective tissues are going to hold us together, provide strength and stability. It's going to give the muscles something to pull against, usually, or at least cover the muscles. The connective tissues between the layers, that in that crinkly stuff you were dissecting yesterday, that's where most of the blood vessels, lymphatic vessels, and so forth run, and the nerves run through that crinkly stuff. And if we will be, when we get to the cats, we'll be doing a whole lot more dissection, and you'll be able to see that really clearly. We kind of skip over that on the rats. Fascia is this tough stuff. A singular form of that is fascia, and the plural form is fasciae. You still say fascia. Anyway, it's this body's framework of these connective tissues, and most every organ is surrounded by fascia at one level or another. I think this is a really confusing part of the chapter. It talks about the three different types of fascia. Superficial is just right under the skin. Deep fascia is when you get down to the muscles. And then the subserous fascia is right below the peritoneum, just right at the membrane on the inside of either the abdomen or the chest. So if you look at it like this, and you're looking at a piece of chest wall here, you've got a superficial fascia, which is right up here in the skin itself. You've got a deep, that's the superficial fascia. It's kind of even got fat mixed in with it. You've got a deep fascia, which is either on top of or below every layer of muscles. And then you have the serous membrane down here, the subserous fascia, which is right above the pleura in this case, or the peritoneum in the abdomen. So those are terms that I think they're not very important for you to try to sort them out. Just know, you'll know what these look like under the microscope, and I think it'll all make sense to you later. Okay, the last two, we have two of the main four left. We're going to cover them in about 5 minutes. Muscle tissue is a very specialized. Muscle only does one thing, and that is it contracts. And that's all muscle does. We have three kinds of it: skeletal muscle, which is the muscles that are under our control, that we can tell what to do and they move us around. The second type is cardiac muscle. You cannot tell your heart to beat. I'm sorry, you can't even tell your heart to beat faster. You can get nervous, in which case your heart will probably beat faster, but you can't tell your heart to beat. It's kind of controlled by itself. Smooth muscle is the kind of muscle that's found in all kinds of other organs, blood vessels, intestines, and so forth. How many of you have thought in the last 20 minutes, you know, I really need my stomach to contract to digest that food I had for breakfast? Right? We don't think about that stuff. It's all automatic. It's what we call involuntary. It's out of our control. And that's what all of these muscles have, the smooth muscle in the wall of it that makes all those things happen automatically for us. Here's what you need to know about the three different kinds of muscles. You need to know which ones of them are striated. Striated means striped. And which ones are not striated. I'll tell you that the striated ones are the skeletal muscle and the cardiac muscle. The non-striated ones are that smooth muscle. It's like in the wall of your guts. Muscle cells can either have a single nucleus. Cardiac muscle has a single nucleus. And smooth muscle has a single nucleus. But skeletal muscle has thousands of nuclei in each cell. Muscle cells can be controlled voluntarily. Well, the only one that's voluntary is our skeletal muscles. The other two, cardiac and smooth muscle, are involuntary. Skeletal muscle cells in our skeletal muscles are very long. They usually go from one end of the muscle all the way to the other end of the muscle. That's one cell. It's also described as one muscle fiber. So fiber means the same as cell when it comes to skeletal muscle. They don't divide. You don't make more muscles. When Arnold Schwarzenegger pumps up for a movie, he doesn't build more muscle cells. He just makes the muscle cells he have get bigger. We can make some new if we have muscle damage, but that's very unusual actually. Here's what skeletal muscle looks like. It's very regular. It has these stripes that I mentioned, and it's found in all the muscles of our body that we control. Cardiac muscle cells, also called cardiocytes. An interesting thing about cardiocytes that you should look for under the microscope over there is these things called intercalated discs. Think about your heart for just a second. And think the way that you want your heart to work is you want your heart to contract all together. You really want every heart muscle cell in your whole heart to contract at the same time, or at least the right time. Okay? So in order to do that, the cells are sort of joined together. The one cardiac muscle cell is connected to the next one by an intercalated disc. And the function of those intercalated discs is to connect those cells electrically, chemically, and mechanically. Think on that for a minute. Smooth muscle cells are the ones that are in your guts and stuff and in your blood vessels. They're very small and tapered. They have one nucleus apiece. Here's the cardiac cells. These are things, this thing here is called an intercalated disc. And just let me tell you, don't get all upset when you go to lab, because you're going to find that on a lot of the slides of cardiac muscle, you can't see the intercalated discs. But on others, you will. It'll be real obvious. Once you've seen it, you won't ever forget it. And I won't try to trick you. Notice also that cardiac muscle cells are branched. And that's another characteristic of cardiac muscle. Here's smooth muscle cells. Each one of these is long and tapered on both ends. They call that spindle-shaped. You know what a spindle is? What's a spindle? Yeah, what?
Wool
I think-
into wool into like thread or some string, right?
Say that again?
Take like a wool or some sort of something and
Wrap it around it?
yeah.
No,
Like Sleeping Beauty, yeah. Okay, thank you. I didn't know. The last one is nerve cells. Just know that nerves, whereas muscle cells only do one thing, and that's contract, Nerve cells only do one thing, and that's carry sparks. They carry electrical impulses. And obviously our nervous cells are mostly in our brain and our spinal cord. We do have some nerves that go elsewhere. Basically, that's what they do. Most of it is in the central nervous system. The central nervous system is classified as the brain and the spinal cord. The other part of the nervous system is the peripheral nervous system, and that's those few nerves, the nerves that are carrying information out to the periphery. Real quickly, most of the neural tissue that we look at is neurons. Neurons are nerve cells, but realize that there are also neuroglia. In fact, there are about as many neuroglial cells as there are neurons. And neuroglial cells are nothing but support cells. They support the brain cells. They make the brain cells work right. Parts of a neuron: cell body, dendrites, axons. Here's a diagram. You can read about this. We're going to go over this in great detail when we get to this section. But basically, dendrites are what bring all the information into the nerve cell body here. This is the cell body. And then there's usually one and only one axon going out, which carries the message on down, where it either connects with another nerve cell or it connects with what it's going to. One last word about inflammation. Inflammation is one of the standard ways that our body reacts to everything. So if you bump your hand, it turns red and gets swollen. If you get an infection in your hand, it swells and gets red. Basically, inflammation is the body's way to respond to all injuries. And what do I think you need to know about it at this stage? I think you need to know that the four parts of inflammation are swelling, redness, heat, and pain. And so you will oftentimes hear that as swelling is tumor, swelling, a tumor, redness, rubor, heat, color, like calories, and pain is dolor. So tumor, rubor, calor, dolor are the four cardinal signs of inflammation. I rode my bicycle across the United States 2 summers ago, and I stopped in a little town in Virginia, and they had a huge memorial to this guy, Stonewall Jackson. Stonewall Jackson was killed, probably by friendly fire, in the Civil War. And God, he's a god down there, I'll have to tell you. I went by and told him I was really glad he lost the war because he was on the wrong side. And then when you go to the South, if you've never been to the South, you got to go because you just can't believe these people are actually living in the same country as us. I love grits. Girls raised in the South. Whatever. All righty. So our next quiz, we're going to have one here in a few minutes when we get over to lab, but our next quiz will be on chapters 3, which is about the cell, and chapters 4, which we just finished, on tissues. Okay? That'll be a week from today. So you've got a week's notice. How's that? Sorry I ran a few minutes late, but we can start lab a few minutes late. Probably don't need to, though, because you got 30 minutes.